First of all, thank you so much for the invitation. I'm really glad to be here. It's my first time here at the EMSL PNL. I've been in Seattle before, but not here, so I'm glad to be around. Thank you also for the opportunity to share with you what we're doing. And one thing that I wanted to do first was essentially more or less connect with what the research, the mission, some part of the mission of the EMSL, and how what we do is directly connected with part of the mission at EMSL. It says that you're interested in understanding enzymes and connect with protein structure and functions. That's what we do all the time. And especially, we are interested in having a predictive understanding. Can we make predictions for biological systems using modeling, simulation, and data analytics? So that's very, very much what we do. I consider myself a computational biologist, but we also have an experimental component in my lab. And whenever we do this kind of predictive modeling, we try to either collaborate with experimental labs or we do it on our own. So I feel that what we're doing is very, very connected to the research interest in EMSL. So I appreciate the opportunity of being here. Okay, so I will start by guiding you a little bit through some of the background of what we do in my lab at UTD. So let's first kind of consider the idea of having a folded protein. So as you know, proteins are polypeptide chains. They fold in 3D structures, right? But they're also under evolutionary pressures, right? So proteins and host organisms actually have a propensity to mutate, change. That's kind of the power of evolution through variation. So assuming that there's already one protein that's folded, is functional, and somehow there's a disruptive mutation or a neutral mutation that somehow tips the balance of stability or functional behavior of this protein. So there are two possibilities. One is that this protein leads to a very unfavorable phenotype that gets discarded through evolution. Another possibility is that after long periods, then we get compensatory mutations that occur very close to this initial mutation that somehow recover an interaction. So this kind of recuperation or this reestablishment of stability actually gets recorded in the history of the family of this protein, right? So here I'm showing you a multiple sequence alignment where these two different proteins, the initial folded protein and the one that has a contact recover, now are part of the kind of history or the phenotypes and genotypes of different organisms. Now, what happens is that this event or this kind of events can be seen as signals that have been stored in the evolutionary record of proteins and their families, and somehow can be used to understand protein interactions, their internal structure, and their function. So if we now look at it from a different angle, so essentially, if we look at this multiple sequence alignment without the knowledge of the structure, so let's assume that these sequences encode for a similar structure, which we know happens a lot. That's why we have protein families. Then we can do some sort of a reverse analysis where we study the statistics of these alignments. We can look at it from the point of view of single sites in this alignment, where we can get probability frequencies, their distributions, and then we can essentially establish, okay, what are the most dominant amino acid distributions at this position, right? People tend to use this a lot of times to understand conservation, right? So if there's a dominant amino acid at certain position, people kind of connect this with some sort of important evolutionary restraint, right? Interestingly, we can also look at the statistics of pairs of sites, I and J in this case, and then we can look at how many times do we observe this co-occurrence, right? How many times do we see an S with a P at position I and J, and N and an I, and then you can build also a joint probability distribution. Okay? Now, the data that we have so far allows us to do this because now we have in the order of thousands of sequences, thanks to genomic and proteomics data sets, and we can use this as first principles to actually build an estimation of the joint probability of the whole family, right? So imagine that now you have measurements for single sites and then pairwise sites, but now you want to compute a probability of all the sites. So P of A1 all the way to L. So you want to know if you have a sequence, what's the probability that this sequence belongs to this family, right? Now, this is a complicated problem because we have an underdetermined system. We have only a few thousands of sequences, but still we can do an approximation where if we use this frequency counts as restrictions in a kind of maximum entropy modeling principle, then we can more or less estimate this joint probability distribution. Right? Now, what comes out of this exercise is a form that looks a lot like a Boltzmann distribution. You have an exponential that depends on pairwise parameters and then local fields, as they're called, or kind of single site parameters. And this is kind of under no other kind of assumptions. This is the best probability distribution that estimates this value. Now, the issue is that now we need to estimate the parameters of this distribution. So you get the shape of the function, but now you want to estimate the parameters. And how to do that, that's a complex problem. These EIJs, what we call couplings, is these couplings for positions and amino acid identities. It's very hard. It's actually a computationally intractable problem. So there's no exact solution, especially for a large L. So let's say an L that is 100 in a typical protein, it's already big enough so that we cannot solve it exactly. So we need to turn into approximate solutions, right? And through the years, there's been several ways to try to solve this problem. Originally, people try the idea of using factor graphs and belief propagation. That turned out to be very expensive and only work out for very small toy examples, right? But some years ago, we tried a different approach, which is kind of based on the assumption that these parameters are really well estimated if you do the inverse of a cross-correlation matrix. So this is what we call the mean field direct coupling analysis. So we are trying to obtain the parameters that tell us something about how pairs of positions in MSAs are connected, right? After that, then there's been other methods, especially in machine learning, trying to use pseudo-likelihood approximations, Boltzmann machines formulations, and sampling techniques that roughly try to estimate these parameters with different kind of solutions, but somehow all related, right? So this, what we realized back in 2011 is that this approximation was good enough to tell us something about the structure of proteins. So what we did learn about this, so we learned a couple of things. One was that whenever we had strong couplings between two positions, I and J, so here represented in below this protein, so we have an alignment, and then whenever we have two sites that have kind of high couplings, we can actually make predictions about the position and its identity with respect to another position, another identity, and then we can assume that these two positions are close by in 3D structure. So this comes from my original slide where I showed that this process of compensatory mutations, that's what it led in the first place to these changes in the alignments, right? Now we're doing some sort of a reverse problem where we're trying to find those locations. We want to isolate this from statistical noise. We want to have this, what we call direct interactions, right? We realize that the top ones, the ones that are really, really strongly coupled, they tend to be in close proximity in the tree structure of a protein. The reason why this was relevant at that time was that it was relatively easy to obtain kind of estimates of local interactions, but far away interactions, long range interactions was very hard. If you were able to obtain long range interactions, that means that you will have a better idea of contact map of a protein and therefore structure. The other important thing about this kind of modeling approach is that we also can see the collection of parameters, this what we call the Hamiltonian, here called HDCA. We can see that some sort of a fitness parameter, a fitness energy or a statistical energy that tells us something about this particular protein, right? And then I'm going to show you some ramifications of this idea. But the key aspect here was that this modeling approach is a global modeling approach. So for many years, people assume independence, right? So the positions in these proteins were somehow independent. Their statistics are independent. But we all know that in reality, it's not true. We have a complex network of interactions that are globally interconnected. So this analysis allows us to obtain this information. It's interpretable in the sense that we're dealing with pairwise interactions. It's a little bit more akin to kind of more physics-based explanations of interactions. Okay, so this approach called direct coupling analysis has had several interesting ramifications. The first one being we're able to predict contacts, right? So the couplings, top couplings in several families show to be close by in the tree structure. If you are able to predict contacts, how two positions in the alignment are close together, if you have enough of those, then actually you can also estimate protein structure, okay? If you are able to predict structure, then why not understanding the dynamics of proteins? So we realize that some of these contacts that were not shown in crystal structures were actually hints towards conformational changes. So we use also this kind of coupling information to obtain multiple conformations of proteins, and we extended this idea also for pairs. So you have two proteins that interact. They are under a similar evolutionary pressure to maintain interactions, right? So if two proteins are changing and their interface is changing, then there's typically compensatory changes that will establish that interaction. So essentially, you can use the same type of analysis to predict complexes or to predict recognition. So if one protein recognizes another, there's typically some sort of a specificity embedded in the sequence that can be analyzed through these methodologies, okay? Coincidentally, right now we are seeing a huge impact on kind of deep learning models of structure and their sequences. We all have heard about AlphaFold, right? And it's kind of impressive results at predicting monomers, and recently also some complexes, right? But what I wanted to stress here is that the principle that really drives this kind of accuracy in predicting structures and complexes is this idea of coevolutionary information. So if you remove the MSA, the multiple sequence alignment data from AlphaFold, you cannot predict accurately proteins. Obviously, there's many more advances in learning from structures, from side chains, and that led to a big jump in accuracy. But the principle of evolution is really what drives these new methodologies, and that's still valid these days. Okay. So in my lab right now, we're interested in the idea of sequence variability, right? So, okay, I have a model of a family of sequences. I know that I can compute the statistical energies. So therefore, I have also a good way to say something about what's the effect of a change, right? So if I know that there's coupling one side that is coupled to many sides, and then if I change that position, what's going to be the effect on this network of interactions? So I can use that predictive power to do molecular design, or I can build models of evolution or understand kind of the effect of mutations in disease, right? So something that I'm going to briefly discuss today is, can you actually build chimeras of proteins where you have kind of lost connectivity and then use this approach to guide design, right? Or can you engineer kind of novel functions in proteins driven by this kind of statistical energy, right? You can build new models of evolution. So since this is coming from evolution, then you can use these ideas as a driving force towards sequence change. I'm not going to discuss this, but that's another topic we're very interested in my lab. How about mutations that affect disease? This methodology allows us to say something about what's going to be the impact of several changes across protein families. Finally, obviously, these days we're very, very interested in novel models of machine learning. So we're interested in investigating new statistical inference models, new deep learning models, but in some sort of a way that is cooperative with this modeling. Right? This synergy actually allows us to say more than just what we've done in the past. So we're excited about this new kind of synergy. And this is essentially what I'm going to discuss further today. How can we merge these two approaches into understanding functional landscapes of proteins and their families, right? This come from evolution and help us understand something about their fitness and their function. Okay. So this is one approach. I wanted to drive you first on how can we use this statistical energy from sequences to say something about kind of the stability or the function of certain protein families. And this is a very neat example because it's an application. So assume that you have a transcription factor, it's a protein that has two modules. One is a DNA binding module on the left, on orange, and then there's another module that is a ligand binding module, right? So that recognizes some.
Margaret was wondering, actually, I was kind of curious about this too. Can you explain the DNA binding model or the DBM, LBM?
Yes. So this is a transcription factor, and then it receives an input from the environment, right? Yeah. It could be a chemical. It binds to a chemical, and then that has some sort of a connectivity that allows this protein to bind DNA. I see. And then that promotes expression or either enhances or blocks expression.
But how do you define the connectivity? From what kind of experiment, or just get it from structural data?
Yeah, so this is internal connectivity. So essentially, you still have a single protein with two domains. It's a multi-domain protein, and it is known from experiment, from biology, that whenever there's a ligand that is bound, then this protein binds to a promoter, let's say. The lac operon is a famous example. The connectivity is not known. That's what we're trying to infer in a way. So we don't know how this allosteric communication happens, but we know that upon binding, this protein is able to bind DNA. That makes sense?
If I think about some of the capability of EMSL, we have several staff who can provide high quality information from mass spec or some kind of omics characterization to provide these informations, but not necessarily come from structure directly. Is that still useful?
So, I mean, it could be, so the more information you have, the better, because then you can see if this model could be also predictive. In this case, the problem was relatively simple to explain. Essentially, what we wanted to do is, what happens if we cut this protein in halves, right? And then we create hybrids. So we mix and match these DNA modules, right? That means that now you can have different inputs and different outputs. But essentially, whenever you create a hybrid protein, what happens here is that you lose functionality. So the protein doesn't work all the time. So people sometimes do this. They fusion to domains, and then they hope that this works.
Does that mean you assume that the binding pocket or binding module is independent of the allosteric transition of the effects?
Well, that's what people interested in synthetic biology hoped, that they were independent.
Right.
So the problem was, hey, I want to create mixtures so that I can use them in some sort of a new circuit, right? But then the problem is that sometimes it works, sometimes it doesn't work, right? And that's obvious, right? Because these proteins have evolved together and they have some sort of a wire connectivity. So what we're claiming is that this method, this statistical energy, is predictive of that connectivity, right? So we show that, okay, the native ones have higher or favorable energies, and then when you make hybrids, they don't. Sometimes they do, sometimes they don't. So that was a previous study. But in this study, what we did is we took hybrids that were not functional, and then we wanted to reestablish those connectivities. And then the way we did is we just look at which changes. We did some sort of a in silico deep mutational scanning shown on the right. And then we show which mutations will increase this, what we call compatibility score. But it's nothing more than that, this energy, this Hamiltonian. So if this was increased, it meant that the couplings were enhanced. So, and then we could try it experimentally, see if there was some sort of improvement. Now you can do this in a sequential order. You improve one single mutation, then you go and then check, the second mutation that will have a good compatibility score, then a third one, and so on. So you could go all the way. So we stop at triple mutants because we wanted to try several hybrids. So different proteins with different DNA binding domains and different environmental sensing domains, as they're called, because they come from the environment. So this is one example where if we see this like operon, that is, it's created as a hybrid with this BSR transcription factor. Originally on the left, that had kind of a functional output, right? So this transcription factor ability, this expression ability of, let's say, 5.9 when we use fluorescence to measure. But then the mutant, only a single mutant that had the best improvement in statistical energy led to a 264-fold increase in this transcription factor ability to induce expression. Okay, so we showed the same thing for different repressors, where like triple mutants on the right, the galactose and per R hybrid actually improved from 3 to 245, and so on and so forth. So essentially, what I'm showing you here is that by kind of reestablishing connections that probably were lost by creating these hybrids, you can actually come back and then design new hybrids that are useful for, let's say, synthetic biology. Why they're interesting? Because then if you already know one particular kind of DNA region that has to be kind of excited, maybe you can use different inputs, different ligands, and you want to create hybrids to do them. So this was in collaboration with professor at UNT, University of North Texas, and one of my grad students, Dr. Chen. This is another example of how we can use this statistical energy. I'm going to go a little bit quicker on this because I want to go into the kind of generative models that are the main topic of my lecture today. But this is another example where there's this family of rhodopsins. So these proteins that sense light, where my colleague from UTD, Shield Odani, she discovered that there was this mutant, single mutant, that will allow a protein to be fluorescent upon chloride binding. Right? So this is a rhodopsin that would only fluoresce when it's bound to chloride. And that has very interesting applications because then it can be used as a sensor. Right? The issue is that this chloride fluorescence was not so high, so it was hard to kind of use it in applications if fluorescence was not optimized. So then what we did is something along the same lines as my previous project, where we look at the family, then we found the residues are tightly connected, and among those were this 121 residue that actually led to this kind of neo-functionalization, okay? So what we can do now is take the top residues that form a network of coupled pairs, and then look at how changes, mutations, in silico mutations could affect its statistical energy. So in this case, because we're trying to go into a kind of a slightly different function, not the ideal function of the family, but a slightly different function, we don't look always at the mutations that strictly improve. We have some sort of a distribution of changes that are acceptable, not destructive, but also not geared towards the original function. So then what we do is then we do mutagenesis guided by this probable interactions that are going to be mutations that are going to be functional, and some of those might lead to better chloride sensing. So what we do is propose mutations, check if the sensors were improving, and then if it's true, then go and then do it again. So this is one example where we took this initial 121 mutation and then kind of using this idea of connectivity and improvement of energy, we selected a new mutant, a new site 132, where we found that mutation E2K led to kind of improvement in fluorescence, and then again we go to the next 84, and then 125, and so on. So at the end, we end up with 5 mutants that actually led to something that actually now could be useful, right? So this is an example where you have control of fluorescent protein on the right, and then our sensor without chloride. And then once we add 40 nanomolars of chloride, then we have fluorescence. I hope you guys can see it online. I think here is clear, but I don't know how it translates on the screen.
So I have a question, Faruck.
Yeah.
So you show us these are 5 successful ones. How many mutants have you tried in total?
So essentially for each of the positions, we did saturation mutagenesis. So about 20, let's say 20 per position. But if you look at the total number of possible changes without knowing where to go, then you end up with hundreds of thousands of possibilities. So that's what we're claiming, that this will reduce your search space.
See, I wanted to point out that EMSL, we have an M2P initiative that has a lot of high throughput screening type of facilities that maybe you and your collaborator would be interested in knowing. I think it'll be very powerful to have your technique and our state-of-the-art ways of detecting phenotypes to combine these techniques together.
Yeah, no, I totally agree, because our point is that these techniques allow you to narrow the search space, but the search space is so huge that we still need high throughput techniques. But at least it becomes feasible now instead of just interactive. Yeah, thanks for that suggestion. I think I agree.
Yeah, I agree.
Okay. So now, let me tell you that all the way we've been using this statistical energy as our guide, right? The issue or the kind of, we want to call it limitation of this energy is that this statistical sequence energy is really one-dimensional, right? I've been talking about favorable versus unfavorable, right? Like if I have a mutation, is this going to lead to worse or better phenotype or stability or catalytic activity? But it's one-dimensional, so we cannot do more than say something about its fitness. So what we wanted to do is we wanted to kind of get, also using the power of learning, we want to see if we could increase this number of dimensions to say something more about not only its function or stability, but also about clustering, functional clustering, right? So we started using a generative method called variational autoencoders. And then the principle is like this. So you have large dimensional input data set. Then you're able to reduce dimensionality into what are called latent variables, here represented by C. And this lower dimensionality is supposed to represent the input data. So the way to do it is that you kind of decode this lower dimensional data, and then you're testing if you can reconstruct the initial data. If you do it successfully, it means that your latent variables are good kind of lower dimensionality representations of your initial data, right? Now, this is useful because we want to have interpretable dimensions, right? So we can work with like millions of parameters, but those are not going to tell us much about the biology. So we want to reduce dimensionality in such a way that is useful. So here, similar to the DCA, we have a multiple sequence alignment that is the input. Then we reduce this into two variables, latent variables, and then we make sure that the reconstruction loss is minimized, and then that the distributions, the probability distributions, this pullback library distance between them is also small. So that's the goal of this kind of learning methods. They have been used also in many other applications in engineering. People use it for images and even to understand molecular dynamic simulations. And in this case, we're using them to understand kind of the sequence space of families, right? Now, what we propose is that people have been using this as kind of like a fancy PCA, let's say. So you reduce dimensionality and then you say something about your input data. What we wanted to do is we wanted to take advantage of one important property of this method, which is that is generative. It means that you can encode your input data, but then you can also generate new samples of your input data that are not necessary in the input set, right? And in machine learning, people use it, let's say, to generate new faces, right? So you encode faces of humans, and then you can generate a new face of another human that never existed. But it has all these properties, right? So we wanted to do something similar here, where we take our multiple sequence alignment, and then we do two different analyses. One, the DCA analysis that I mentioned before, the DCA, and then we also encode and decode with the variational autoencoder. We use then this generative property where as soon as we have this low loss, then we can create these plots as you see them here. There's a 500 by 500 grid where each pixel in this landscape is a sequence. It's a generated sequence. It's not a sequence that is found in nature. It's a generated sequence by the model. And then for each of these sequences, what we do is we quantify it with the Hamiltonian, the energy that I described before, right? Because we know from this energy that we can say something about its fitness. Say. So that's the color, it's the Hamiltonian that comes from the CA. The X and Y axis are these two latent variables, right? So now our input are many sequences or output are 250,000 sequences that are created by the model. And then we create this map where we can, again, plot back the initial input, the initial extant sequences, so sequences that are real, and then see where they land in this map. So here I'm showing you an example, hemoglobin, and then we have subunit alpha in green and subunit beta in yellow-orange. And we see that we have these very interesting structures where we have kind of dark regions that are favorable regions, and then we have light regions that are unfavorable regions that we see as barriers, right? So you start having these structures that you do not get if you don't quantify each of these sequences. So if you just plot this family just with the VAE, you would think that it's hard to point, but let me see. You will think that these sequences, can you guys see the?
Yeah, they can see.
Yeah. So these are close by in terms of the X, Y axis, but they're far away in terms of a barrier that has been here. So things that are more similar, they tend to be in these local kind of deep or bubbles that are in this landscape, right? So that's something that you're gaining now. You generate sequences, you quantify them, and then you can start saying something about their families, right? In this case, for instance, I'm showing you this hemoglobin family, and then we see that these maps really cluster really well in terms of the phylogenetic relationships of these proteins, right? So you see that they're all kind of separated by these barriers, and we have a clear distinction mostly of this different kind of phylogenetic clusterings, right? Now, we also ask the question, what happens if now, instead of looking at the natural sequences, what if we sample from those kind of non-natural sequences that were created in the model, and then see if they will maintain a phylogenetic relationship similar to the input data? So in this case here, in the top plot, we have essentially, we compare trees. So this is a cluster information distance. It's a way to compare phylogenetic trees. So we take samples from the same extant sequences, natural sequences, and then we see how they're different, right? So since they're both part of the same family, we end up with a distance that is about 0.4. That's typical for two samples from the same family, right? Now, if we sample them, so now if we extract, if we generate new sequences, and then we compare them against globin, we have roughly the same distance. It looks like we are preserving these phylogenetic relationships. In contrast, when we compare the globin, let's say, with a different family, the saturase family, then we see a much larger distance of 0.8. Or if we basically compare globin versus a shuffled version of globin, essentially a random protein, then we see like a distance close to 1, right? So what we show is that even though we are generating sequences that are not in nature, they do preserve this phylogenetic clustering, which is very interesting. Okay. So what else did we found here? So this is another family, beta-lactamase, and again we see this kind of clustering based on phylogenetic and functional properties. But then we see some other interesting things. For instance, if we zoom in here and then we look at different projects that dealt with these beta-lactamase proteins. So these proteins are antibiotic-resistant proteins, right? So they very cope antibiotics. And then we see that, for instance, there's these two subfamilies of beta-lactamases called TEM beta-lactamases and SHV lactamases. They're very similar. Their tree structure is almost identical. But here we do have a very nice separation on the right, where we have the two different members. And it might be hard to to see online, but there's some sort of barrier separating these two. Right? Now, another thing that we observed is we took two different experiments where people actually took one lactamase, then evolved it in vitro, and then they were experiencing mutations, high mutational rates, and then they basically only recovered those that survived. So this in vitro evolution experiment, and we saw something very interesting on the left-hand side. Sorry, on the right-hand side, Fantini et al. What they did is they had a really high selective pressure. So their antibiotic was concentration was really high, and then we see that all the sequences that are coming out of that experiment, those selected sequences, they tend to go into this well, right, of functional space. But then on the left-hand side, this was an experiment where the selection pressure was not high. And then the sequences tended to go over, if you see, they go over places with barriers, right? So their amino acid identity was much larger, but also they were under less pressure. So this is captured by this map really well.
Faruck, are you pointing on the screen, because I can't see it online? Are you, like, for example, saying left? Which one is the left and which one is the well? Can you, like, just use your mouse to?
Let me use my mouse. Yeah.
Okay, thanks.
So
I think I got lost, because a lot of things you're trying to teach us how to read this diagram. But
yeah, I will stay here and use my pointer. So this is the area where we have high selective pressure, and if you see, most of these variants landed in this well that has like a more favorable Hamiltonian, darker Hamiltonian. Well, this experiment, the selective pressure was low, and then the variants that were obtained landed on barriers. That's an interesting observation, where these sequences are not supposed to be very selected for high concentrations of antibiotics.
So when you say Z0 and Z1, can you just quickly go over what that is? Because I think I got lost when you're trying to explain this diagram.
Yeah, so there's no, let's say there's no physical or biological kind of one-to-one mapping of this. So these are latent variables. So these are variables that have to do with learning. So what we're saying is that one coordinate defines a protein. So a coordinate C0 and C1 defines a space, and that space has some properties with respect to the actual family, right? So where we plot our families, this coordinate system is allowing us to separate things. So it's like a clustering procedure, but the color, that comes from the statistical energy, right? So we needed these three variables, these three dimensions to say something about this set of sequences, right?
I see, but isn't that your VAE is supposed to be interpretable? So what are those? Can you speculate what underlines those latent parameters?
Yes, so our interpretation is that these clusters represent one, phylogenetic or functional clusters. So these are sequences that will behave similarly, like in this case, where we have sequences that bind to SHV. So these are SHV beta lactamases, and these are TEM lactamases. So these are both sequences that bind to antibiotics, but they have slightly different properties. One has a wider binding site that allows larger antibiotics than the other. So these coordinates are able to pick that, even though in sequence space, they might not be easy to distinguish them. That's the point.
Ah, that's great. Thank you. So which means that the latent information is embedded in a three-dimensional structure.
Yes, three-dimensional structure or the functional aspects, catalytic activities, and so on. And what I'm trying to show you is different examples where we're observing these properties. Yes, that's correct.
Thank you.
So this is another example where This is a TRMP8 protein. So this is a sensor. It's a transport protein, and it senses temperature. So it's a cold-sensitive protein. And there was this very, very interesting paper, Matthews Cruz et al. What they did is they realized that squirrels that hibernate, they have a variant of this protein that makes them less sensitive to cold. Well, rats that have their TRMP8 version, they're sensitive to cold. So they wake up, they did experiments where they realized that, okay, rats are much more sensitive to cold. So what they did is they essentially took the squirrel version and then put it in rats. And then they realized that they start losing cold sensitivity, right? Now, because our view is that this protein, its main goal is to actually be sensitive, not insensitive, then the squirrel versions of the protein are really in this dark spot, right? So that's where you have the, the rat, I'm sorry, the rat wild type is here in a very dark spot. But then what they did is they did mutations that allow the protein to lose sensitivity. And then that's where you have this rat6 mutant that is not sensitive to cold. So if you see it goes out of the well and it starts going into a barrier region in the landscape. Right? Same thing with the wild type of the squirrel. The wild type of the squirrel is not really, if you see it here in 3D, it's not really down here in the well. It's outside, it's up there. And that's why this protein is not that sensitive, right? Then you could still do changes in the squirrel to bring this mutant to sensitive regions, right? So in this case, what we're trying to show is that a good representation of what a functional protein looks like can be encoded in this kind of three-dimensional structure. If you see it only with the latent variables, you will not see this difference. You do need this third dimension, which is the Hamiltonian. Okay? Okay, how much time do I have?
Probably just a couple more minutes because it was almost like...
Yeah, I'm almost done. So I'm just going to skip the next example, which was kind of a similar. But this is something that I think is very useful and you might find interesting. You could also use these maps as a way to predict or annotate function of proteins. So essentially, if you have your map like this, and then you have a new protein for which you don't know what's its function, and it lands here, then one really easy way to predict its function could be just to relate it to the neighbor. Right? So you predict that this new sequence that lands there might have the same function as its neighbor. So that's a very simple way to predict function. And we implemented something similar to that, where we will take many, many sequences that were not annotated, and that then later were annotated, and we check how many times did we make a mistake. And we then compare to other kind of state-of-the-art techniques using language models. These days, language models are very, very kind of popular. And then we realized that just by doing this proximity, we can do a better job, or at least as good as a job at predicting kind of function of this protein just by the location in this landscape. Right? Here, I'm showing a comparison against this language model, and then we see that LGL, VAE, that's our method, and we see that NLM is a language model that does something similar. But then you notice that there's this region called coverage error. What we call coverage error is the system will give you a name that is convincing, is not incorrect. But here's an example where this was an aspartate tRNA ligase, but this had aspartate and also aspargine. But then their method will just give you aspartate tRNA aspartate ligase. It will miss the aspargine ligase. So that small specificity is important, right? Because this has dual specificity, but the other method will not find it. In our case, because we're just kind of replicating the closest neighbor, that's why we got those hits correctly predicted. So as a way to annotate novel sequences no one has been analyzing, this is a good approach too. Okay, and yeah, so I have a couple more examples, but I can discuss those later. This have to do with evolution, people trying to, let's say, convert a beta hemoglobin into an alpha hemoglobin. They did experiments, and then we show how these experiments essentially show a crossing from the barriers from the beta to the alpha, and then those that are in the barriers were not functional, and only when things close barriers, then you start having ancestors that look like alpha globins. Okay, and let me just finish by showing you an example of coronavirus, where we did this with the spike protein, and then we show very clear separations between what is sequences of SARS, human MERS, SARS-CoV-2, and then right in the middle, we have this bad VCV sequence that some people believe it could be the link between the kind of the human version and the animal version. And it's just there, crossing the barrier, getting into this region of SARS-CoV-2. And we look at all the different variants, and then we can see the progression, how one variant is all clustered together, and then suddenly new variants start appearing, and they start forming kind of their own slightly different clusters. First to the left, where you have the Delta variant gets popular, and then all the way to the right, you have the Omicron variant. So they, even though they're all very similar, they still have these slightly different properties that are reflected in the landscape. Okay? So yeah, let me end up with that, just by saying that we think that these generative models are useful to predict function, organize families by fitness, to annotate them, and some interesting application, it's also for protein design. Because in theory, we can take sample from our landscape and then design a protein with those protein properties that might not exist in nature. Okay? Yeah, I just want to thank my students. This is the building where my lab is, and my collaborators, Sheil and Clement, and my students. Thank you for your attention, and also I want to acknowledge grant funding.